we are trying to solve. Yeah. So this is a good opportunity to learn at least know few more people, right? So that after the workshop you can start keep talking to each other. Yeah. Okay. So after the before the lunch session, so you will have.
I will come around 1 o'clock here.
Okay.
My colleagues are here. If anything is in between here, then
Yeah.
the soft copy distribution, if you want to do it, we can.
Yeah. Okay. So they are in good hands. I'll take care of them.
Yes, sir.
I'll entertain them for the next 3 days. Yeah.
Yes,
Yeah. Very good.
I will come around.
Yeah. Yeah.
I will come.
Yeah. Perfect. So I'll leave this slide on before I leave for lunch. Yeah. And what I want you to do is write down in the next 5 minutes or so on these things, and that is so, and give that piece of paper to me so I go home and read. Yeah. So that's my homework, and then I'll give you homework so that you do some presentations tomorrow. Yeah. Okay. So anyway, I love this slide. Yeah. Life is really beautiful. It is amazing, what life can do. So there are these scientists. Have you heard of Carl Woese? Yeah? What have you heard of him?
He proposed 3 domains of life.
That's right. So this was an amazing contribution, doing something very simple in fact, right? So he proposed that there are, so before we used to call prokaryotes and eukaryotes, yeah? And that's, so let me ask, so what does prokaryote mean to you? Why were they calling it prokaryote? Anybody? No, only when your turn comes, you speak, yeah? So yes, prokaryotes.
Prokaryotes are early form of living creatures which don't have organized nucleus.
Right. So the karyo stands for the nucleus, right? So the mindset was before that there are things, because they could see nucleus in the human, mammal or plant cells, and that wasn't visible in the bacteria. So they started calling them as prokaryotes and eukaryotes, right? Without and with nucleus, yeah? Because they were just trying to divide or understand life. So the contribution from Carl Woese, and so have you heard of this guy, Norm Pace? Huh? Anybody? So I'll talk about him. So he also made a huge contribution to this as well as other things, and he's a professor in my department. So based on Carl Woese, then they said, it is bacteria as one domain, then archaea and eukarya. Yeah. So that's what proposed for the 3 domains of life. And I have copied for you exactly the same thing, but just presented differently. Yeah. But my question for you is, what's wrong with which of these figures? So 15 seconds to think. If you want to say something, raise your hand. Hold on. All right, anybody from the second half of this room?
Archaea.
What's wrong? Archaea?
Archaea.
What's wrong with archaea?
Archaea is a bacteria, then archaea, then eukaryote. But it's like the lineage of archaea coming with eukaryotes.
So which part is wrong? It is this part or this part?
This one, sir.
This is wrong. Okay. So this part is wrong.
Usually it comes with bacteria.
Okay. So those who agree with our friend that there is something wrong, with this here, raise your hand. Yeah.
Actually, I think that.
And speak loudly so they can hear. Yeah.
I think that the first one is non-anchored, means it don't have its origin species, and the second one is in anchored, means ancestor species is
So if I were to look at this, yeah, I might have get an impression that probably this is the root, yeah, the starting point, and out of that more complicated things came, and maybe there was this one lineage here and one lineage going this side, yeah, and that is an old mindset, yeah. So this is a, you know, much better representation of that idea, because what you will see is, so we in fact will put over here this word called LUCA, LUCA, yeah. Do you know what LUCA is? Anybody of our friends over here? LUCA. Last universal common ancestor, right? So the point is that there was something before, yeah, from that came a point in life where things split, yeah, and what you'll see from here is that this is presented over here, and what this implies is that this has something which are common to both this group and this group, yeah, and on certain ways these are, because before when the prokaryote and eukaryote mindset was there, that time it was that there are things, you know, before the nucleus and after the nucleus, yeah. But what this is saying is this is a third independent domain of life which has some similarities to this group, and there are other similarities to what's in this group. How many have heard about the RNA world? So our okay, and speak loudly, as loud as you can, yeah.
The RNA word is basically highlight the importance of RNA, because like basically for DNA we talk about genetic material.
Yeah.
For RNA it has the genetic material and it has the enzymatic function also.
Perfect. So I am from University of Colorado. I hope you heard that part, yeah. Now there is a guy named Tom Check or Thomas Check. Anybody have heard his name? Tom Check or Thomas R. Check. So he got Nobel Prize in 1989 for the work he did in about 1983 or so, yeah. And he got Nobel Prize for the discovery of ribozyme, yeah. And ribozyme is that RNA is an enzyme. So until that point, our biochemistry and molecular biology textbooks used to say that all enzymes are proteins, yeah. So with his discovery, we have to literally rewrite those books, because what he said was that RNA can also be an enzyme, yeah. And why was that Nobel worthy or Nobel Prize worthy discovery? It was because until that time there was a chicken and egg problem, and the chicken and egg problem is that who came first, chicken or the egg, right? As we know, chicken comes out of egg, right, and egg comes out of, yeah. So the question is who came first. So we knew for a long time that DNA is the genetic material, yeah. Good so far. And we also knew that proteins do the stuff in the cells. Proteins are the enzymes or the machinery to do the stuff, whether you lift your, you know, something from the table, or think in the brain, or your kidney, you know, clears things in the system, yeah? So there are proteins doing the stuff. So now the missing link in between was RNA or the mRNA. So that's where Crick, you know Crick, Watson and Crick guy. So Crick proposed that there has to be some intermediate in between, because DNA is made up of 4 things, A, G, C, and a T, and proteins are made up of 20 things, yeah, and they are unrelated to each other, yeah. So now the conundrum was in the 60s and 50s that what's happening inside the body, right? So anyway, it was required that there has to be a messenger, yeah, which takes information from the DNA and now somehow makes proteins out of it, yeah. That turns out to the messenger RNA, and so on. So the proposal of the discovery of ribozyme was that it's supposed to have solved this chicken and egg problem in biology, and that was, if DNA is the genetic information, proteins do the stuff. Yeah. So which came first? Because proteins can't replicate, proteins can't divide. Yeah. DNA can't do the stuff. There are some exceptions we can talk about. So RNA, when proposed to be as an enzyme or ribozyme, so it solved the two problems. So as if, so now I want you to kind of mentally go from this part, that's where these are in cells. Yeah. So there was a life if we go a couple of billion years before that. Yeah. So what happened around here was that probably, that's a hypothesis, that probably way back when life, which we call as life is beautiful, it was trying to start, it might have started as RNA world. Yeah. Because RNA could do both the things. It can be a source of genetic information because you can copy it, because there are lots of RNA viruses. Yeah. So you can make RNA out of RNA. You can also make DNA out of RNA. And what they are saying is RNA can also do stuff, can have catalytic activity. Yeah. So it is possible that way back when over here, yeah, that there was an RNA world where RNA was doing both the things. Yeah. So my connection to, unfortunately, the plant world stopped when I left IRI. Yeah. But plants are still near and dear to me, and I think to the humanity. So this slide will come back on the last day, and I'll put that as the last slide of the whole presentation. And that is where I'm going to put all of the burden on your shoulders. Yeah. Because I will ask you, so what you think are the problems in crop sciences, agriculture, or in this area, and what do you think are the solutions, what you can do about them. And the point will be then how can we use either computational biology or next generation sequencing to do things in a much better and faster way in either these problems or these issues or something that you may have your own ideas. Yeah. So you have between now and Friday afternoon to think about. Good. So we'll come back to this. So anyway, so we know that there are a few things which are quite important. That what you want in life is that you want more output from the limited or the less input, right? That's very good. And this plays a quite important role in the plants. And in fact, when I was a master/PhD student, so I was talking to Professor Sushil Kumar. Anybody heard of his name? It's okay if you don't know his name. So he was a bacteriologist, and I made a stupid comment to him, and literally it offended him. And I said, so we know that legumes can fix nitrogen, right? Cereals can't do it. Yeah? So the idea is that what if you dream one night and find that you can make cereals also fix nitrogen? Yeah? Good for the farmers. Yeah? Because there's plenty of nitrogen up there, they can do the same thing that. So I told him that I don't think that's possible or feasible, because if it was possible, then nature had billions of 4 billion years to take care of it, but it never did. So there must be some big barrier to that. Anyway, I was scolded for that. So the point of that was that don't limit your thinking. Yeah? Because you can do certain things that hasn't happened before. Yeah? So keep that option open in your, keep your mind open. I was told that this is a big problem, that farmers are applying more fertilizer than that crop needs at the time. So what happens to that? Just like we had rain 2 days ago, and I was told we'll have rain tomorrow, and I can't believe our weather forecasting has become so good now. Back in the, when I was growing up, the weather forecasting was all about यूपी, बिहार, राजस्थान, दूर दूर इलाकों में जोर से बारिश होने वाली है, right? या छींटे पड़ने वाले हैं, right? And I think as long as some छींटे somewhere, right, their prediction has come true, and so on. So anyway, so this is, I think, a problem. And again, you as scientists have to somehow connect to the farmers, right, and get the message across, because this may end up the same problem that we have with, say, antibiotics, because now there's an overuse of antibiotics, and we are having problems with that in the society. And unfortunately, this can also depend on our friend act of God, right? Wrong time, wrong baris makes a problem. And I'll leave the rest of it as it is for you to think about. But anyway, we know there's a lot of potential and scope. Yeah? And as I was trying to point to, that this plant breeder will take about 10 to 12 years, yeah, to improve whatever he or she wants to do in any crop. Yeah? So the point of this genetic engineering or molecular biology or computational biology is, can we speed up the process? Yeah? And that's a big deal. So again, put that thought into it as to what problem that you would like to solve and how. Yeah? So that when we meet, let's just say again 10, 15 years from now, we say that, we thought about that, we did, and it worked. And I think, so I'm sure that you have heard a bunch of few examples of what happens in here. And this is simply to say it's a nice, juicy, red tomato. Yeah? And it's very good. But the problem is that, the ripening makes a big difference. If you transport things from place A to place B where consumers are going to use it, in between, there's a lot of loss. And that happens to, so I was growing up in a village about 100 kilometers from here. And what happens is that you harvest a crop, say in April, then summer life is good, it's very hot, but come July and August, there is so much moisture, so much humidity, and there's a problem for the seeds. So I happen to be I did my MSc in Seed Science and Technology, yeah, and I was the first one to make a history in IRI that I was probably not a good student, that one MSc wasn't good enough for me, so I was the only student to do a second MSc in IRI, and after that they made a rule that you can't do it, anyway, but it's okay that you made history, and so on. So anyway, so we'll come back to this slide at the end of this, right, and then see how do we connect these dots. Good? And I'm going to next make a case that even though my research directly in the last whatever years on agriculture, but what we are going to talk about applies to plants and animals exactly the same way. So the whole point of going around the room was, so I get a sense of what you are from, what background you are from. And if I have to describe in one word that what I heard, that this is a diverse group, right? So now the challenge for me is, if I only cater to someone who already knows a lot of genetics, or someone who thinks he or she knows something about computational biology or so, then the other people are lost. Yeah. So anyway, my point is, if I wish to be a politician act, sabka saath, sabka vikas. Yeah. So it will only work if I'm able to bring you all up to speed, right, and challenge even the most, knowledgeable in this field. Yeah. And what we will do is we'll kind of use you, right, in our that practical exercise, that you are paired with some people who has little bit of either you know more about that, so that they can help us out. Yeah. All right. So how many of you know this? And I have prepared, and I was in the darkness because I did not know what I'm getting into, right? So at least I have over prepared. Yeah. But the point is, on the fly I can delete stuff, or I don't happen to see a blackboard here, but I'm good at using blackboard, but we'll figure something out. Yeah. That whatever you have, whatever I didn't think about, I can use some other ways to interact with you. All right, so you all know this stuff, right? And that's a very good thing. Now look at it carefully. Did you see anything change? Yeah? Okay. So what is happening there? It's just so subtle a change. Okay, here. So this is the first, this is the second, and what's wrong with these flips? So our DNA, which we call is normal B form of DNA, that's which-handed? Those who did not know before. Huh? Right-handed? Yeah. So this is our friend Watson and Crick. So the Crick of that Watson-Crick fame told us that genetic information flows from DNA to RNA to protein. And if you look at this figure has been given more Nobel Prizes than any other figure in biology. So let's just go through just to bring some of you up to speed. So who got Nobel Prize for this? Anybody from here? Anybody from this row? Remember, I said for this arrow. Say loudly. I can't hear it. Hold on, just from here. So what is called the father of DNA replication, and there is only guy who fits that bill, father of replication, Arthur Kornberg. Yeah, because what Arthur Kornberg did was, so he was able to purify things that you need for DNA synthesis in a test tube, and now he can mix and match everything, and he told us everything about what you need for DNA replication. Okay, how about this arrow over here? And only this row. And there are many, you can name any one of them, and I'm good. Say loudly, please. David Baltimore is wrong. Sorry. Huh? So Roger Kornberg was one guy, and he got a Nobel Prize for the solving the structure of the transcription machinery. Yeah, and this guy Roger Kornberg is the son of Arthur Kornberg. Yeah, so that's strange. It's very rare that father and son get the Nobel Prize. Okay, how about this arrow? Now you can say David Baltimore. But there was, I'm not going to ask you that question now. The question is, who, in addition to David Baltimore, got the Nobel Prize for this discovery? And anybody from this? Huh? Howard Tamin, right? So this was, again, this arrow didn't exist in our heads. So they said they discovered in a what is called retrovirus, an enzyme called reverse transcriptase. And they said that is what is used in the cells to copy RNA into DNA. How about this arrow? And give me a name other than Hargobind Khurana. My friends over there. So Khurana, Marshall Nirenberg, and Ochoa. Yeah. And there was a Nobel Prize given. There is no arrow over here, but suppose I were to put over here. So there is a pseudo fake arrow, yeah, which is why I didn't put that arrow. But suppose there is an arrow over here. Who got Nobel Prize for that? Who didn't have a chance to? You guys. Huh? So the guy named Stanley Prusiner, the guy who discovered prions, this mad cow disease. And why I say it's a fake arrow is because normally RNA can be replicated, yeah? DNA can be replicated, but there is no such machinery which can replicate protein into other protein. But what he said was that if suppose in that prion a protein gets bad, yeah, it changes into a conformation that can now change the neighboring protein into bad. So it can now have this dominoes or cascading effect. It can make everything else bad. So anyway, then they said, and that is heritable, meaning that you just need that one copy of bad guy, yeah, and it can, when the cell divides, that one is enough to turn all the other good copies into the bad copies. Yeah. So I still call it a fake arrow. But if you can find a real arrow, I can guarantee you, you will get a Nobel Prize. Yeah, so discover a machinery that can replicate proteins, and you are guaranteed with the Nobel Prize. Yeah. How about this arrow, the going backwards? I didn't show an arrow here because none exist. Yeah. So there is no machinery that I'm aware of which can take a in the cell, which can take a protein and convert it into the RNA. Even though in computer we can do that. The reason we can do that is we have this, our genetic code, yeah, 64 of those guys, 3 of them are stop codons, 61, they correspond to one of those 20 amino acids, right? So now, if you know the sequence of nucleotides, you know exactly the sequence of amino acids in the protein. But the backwards is only partially true. That suppose you know the sequence of proteins, yeah, you can try to come up with a sequence of RNA, but remember, the genetic code is degenerate, and because of that, you will know exactly which one, because there are 6, like this, 6, right, codons for arginine. So one of them could have done that. You do not know which one of that is, right? So again, the point is that if you can, in the next 20 years of life, come up with a machinery that does this, you will get a Nobel Prize also. Now you can't complain, you were not given this idea, bright idea how to get a Nobel Prize. If you don't get it, you didn't work hard enough. So this is just for the friends, who are from not necessarily the molecular biology field of it. So transcription from the previous slide is very important. In bacteria, what happens is that we have our friend RNA polymerase that's going to transcribe this DNA molecule, and RNA comes out. And remember, DNA synthesis happens in a 5' to 3' direction. So is true with RNA synthesis. It can only be synthesized in a 5' to 3' direction. One of them will become a template strand, so this RNA is complementary to that strand, and because this template is complementary to this sequence, RNA is also complementary to this sequence. No, RNA is also complementary to this sequence. Therefore, the sequence of this is exactly the sequence of blue. Good so far? Because they are both blue and green complementary to this brown strand. Yeah. The only difference is this has ribose sugars and this has U instead of, very good. So in bacteria, yeah, and I think that's probably relevant to the people who are from the mycology and plant pathology. They deal with these microorganisms. So bacteria has a bunch of genes, and now the question is, you do not transcribe all the genes all the time. You only transcribe some genes at certain times, depending on what you needed. So the question is, how does the bacteria accomplish this task of only synthesizing or transcribing the right kind of genes? Pardon? So how many of you have heard of the word sigma? Okay. So to those who didn't hear of sigma, so sigma is so our RNA polymerase comes in these two forms, what is called the core enzyme, and core enzyme is the one that knows how to synthesize the RNA. Yeah. But the problem with the core enzyme is it can bind to DNA but very loosely, and second is it doesn't know what is the right place to bind. So it just loosely binds everywhere. Yeah. So it has no specificity. So what sigma does is that when sigma binds to our polymerase, it makes a holoenzyme, and that holoenzyme because of sigma, it knows exactly where to bind, and we call that where to bind as our promoter sequence, -10, -35 sequences. So that's the binding site for the sigma factor. So kind of think of the sigma as if I were blind, yeah, then I need that walking stick. So think of our sigma as the walking stick for the core polymerase, because it is going to guide the core polymerase to the right place where it needs to go. And once it does that, it now has a very high affinity for binding, and it also binds to a very specific place. It tells the core where to start transcription, yeah, because that's key. If you just start transcription everywhere, it is not going to come out very good, yeah. And this is my favorite sigma 54, because that has to do with nitrogen, something with nitrogen. So what happens is there are these housekeeping genes. Housekeeping genes are that you need them most of the time, yeah. Say how to deal with the energy, yeah. If you are given a glucose, how to deal with the glucose and so on. There are also, so you need certain protein enzymes to be made, but there are other things, and these are only expressed when you are starved, meaning there is not enough food around, or when you are heat shocked, yeah, or when you are problem with the nitrogen, enough nitrogen in the cell or not. So these right kind of genes are synthesized in response to those conditions for the bacteria. And now there is a specific sigma for each of these conditions. So for example, this guy sigma 32 is for heat shock, because the cell needs to deal with the heat shock. When you give a heat shock, some proteins are misfolded. That's very bad for the cells. So you need to have some way to deal with that. So what sigma 32 does is, it is synthesized in response to heat shock. It now binds to the core polymerase. So this friend at the very top called sigma 70, so it binds to a sequence of this type, -10, -35. Yeah, those are the genes that you normally need. Yeah, when sigma 32 is absent, but now you give heat shock, sigma 32 is made. It now competes for the sigma 70. That's our housekeeping sigma or the vegetative sigma. So now this sigma will associate with the core polymerase and tell core polymerase to go to those promoters and transcribe those genes, yeah, which are there to deal with the heat shock. So the question for you is, how do you think the heat is sensed? How do you turn it on? Speak loudly so I don't have to repeat it. And there is also in the sigma 32 RNA sequence, there is a stem loop, yeah, and that stem loop, as we know, RNA structure can change when you have high or low temperature. So that structure hides our translation start codon or AUG in that structure. So suppose this is the AUG, which is our translation start, and this is part of this RNA structure, yeah, and therefore translation machinery cannot get to it and cannot translate. Now comes heat shock. It is enough going from say 37 to 42 degrees, right? Then now this opens up. Remember, RNA will unfold when you heat it up, yeah. So this is what happens, and now the ribosome can get access to that and start translation, just as one example, yeah, and so on. So anyway, the point here is, this is to give a flavor of that depending on what genes you will need, yeah, there are specific sigma factors. They will reuse the core polymerase and allow it to transcribe right sets of genes depending on your needs. And this is simply an example of that if you look at the percentage of total transcription when you increase the heat shock, yeah, so these are the genes which are sigma 32 stimulated, yeah, and these are the genes which are sigma 70 stimulated. And the question is, based on this data, anybody in this table, what can you conclude? Next, I'll come to you guys if they give up.
So there is inverse relationship, like when osmotic shock is increasing, there is expression of sigma 38 factor. In the reverse, it is the sigma 70. Their expression will reduce at that time.
Yeah. So the point to get here is that when you do this osmotic shock, and this is dedicated to these kind of genes, so then these sets of genes are, in terms of the total, are now more and more transcribed, right? Because these are right kind of genes. Now, how do you see that this sigma 38 is competing with sigma 70? Yeah, because then the opposite should happen to the other housekeeping genes. And what you can see here is that indeed these our housekeeping genes allowed by this sigma, now transcription goes down. Yeah, so that's a switch. Yeah.
Is there a possibility that sigma 70 has interaction with sigma 38? In presence of his lower expression, that expression is high.
So these are the genes which are transcribed by sigma 70. So those genes, their transcription goes down.
If accumulation of 70 is less, then expression of sigma 38 is
Accumulation of the mRNA for the genes which are responding to sigma 70 is less. Yeah.
is less,
Yeah.
then expression of sigma 38 responsible mRNA is high.
Is high. Yeah.
So they may be negatively correlated.
Right. So those are inversely correlated, right? So now the question is, but that's a good point. So let's just say that you are born 40 years before you were born, yeah? And this is where the field is, and you want to figure out what's the mechanism of how it happens, yeah? So what experiment will you do? So there are two possibilities I can think of. One is that, as I said, so what happens is, suppose this is the core polymerase, yeah? And this is my sigma 70. When it binds to sigma 70, it transcribes all the housekeeping genes, yeah? Now, in response to osmotic shock, this sigma is turned on. So what happens is, so now all of those genes are not transcribed, but these are transcribed. But now the question is, I have made this up, yeah? But suppose you want to actually establish scientifically or experimentally, what experiment will you do about what's the mechanism? That how it is. So one possibility is this, yeah, versus this. Other possibility is, how about these two talk to each other, right? This maybe inactivates this guy, yeah? So how will you distinguish these two possibilities experimentally? Say loudly, otherwise I have to repeat to your friends.
So constitutive expression of both, like if we allow to express both at a time.
Yeah. So one is, so what will you find if you allow constitutive expression?
So we can find, like if it is not varying, there is no variation. So then we can think that both are being expressed at the same time. Or if we find the variation, then one expression is affecting the other gene expression.
So suppose you do an experiment that you can put now sigma 70 also under the osmotic shock promoter, right? You turn it on, yeah? Then what you are going to find is that instead of these coming down, yeah, they will either not come down or they will increase just like these, yeah? But that will not distinguish between this possibility or this possibility, because in either case it still remains valid. So let's just go to your idea of protein-protein interaction. So what will you do?
So the way we can go for co-immunoprecipitation.
Co-immunoprecipitation, right? So let's just say you have antibodies for your core protein, RNA polymerase, and you do immunoprecipitation, and suppose you have antibodies for both sigma 70 and sigma 38. So what you do is, you take your cells, osmotic shock, so before and after, collect the proteins, and now immunoprecipitate using antibodies for core polymerase, right? And now do a Western blot. How many of you know what's a Western blot? So those who don't know, so Western blot is, you isolate proteins, run it on a gel, separate them based on size, transfer to a membrane, and now you can probe that with antibody, meaning detect that protein with an antibody. So what you are going to expect to find there is that under this condition, when you do the co-immunoprecipitation, the amount of sigma 70 coming down will be less, yeah, upon osmotic shock, whereas the amount of sigma 38 coming down will be more. So that means both are interacting with the, and what is happening is that one is displacing the other guy, because if they are two together, yeah, suppose, then what will happen is that sigma 70 both before and after will remain the same. Yeah, so anyway, you can distinguish experimentally which of the model or mechanism is correct. So again, just to bring you all up to speed on gene and gene regulation. So if you look at very distantly how genes can be regulated. So we talk about that you can have either a negative control or a positive control. So this is our friend RNA polymerase. So normally it binds to that promoter sequence and turns on the gene. But suppose there is a repressor made, so that competes for the binding of the polymerase to that promoter sequence and doesn't allow polymerase to bind, and therefore no transcription. Yeah. So this is example of negative regulation, that a repressor is preventing polymerase from binding to the DNA and no transcription. Also it is true on other genes you can have a positive regulation, and that means that there are these promoters where RNA polymerase doesn't bind well, doesn't bind strongly. So it needs some help, and that help comes from what we call as activators. So what an activator does is it helps polymerase bind to otherwise weak promoter, the weak genes, yeah, and therefore allows their transcription. Anybody know who these guys are? Anybody from here? The operon model, right? So these are literally the architects. So if you are in the 1950s and 60s, yeah, and you are trying to understand what happens inside the bacteria, so they did some absolutely simple but wonderful experiments, and that's why they got Nobel Prize for their work. Jacob and Monod for operon. So I'm going to cover this operon in probably next 35 seconds or a minute. So what's the key thing about the lac operon? So after all of that, you cannot complain that you are not from the molecular biology field, yeah, because everybody should be now up to speed after this. And for some of you, it sounds a boring discussion because you know so much, but just bear with me. Okay, here. Anything about lac operon? What's key about lac operon regulation? Hold on.
Sir, many genes are controlled by a single regulatory molecule.
That's it? Okay. So bacteria can feed on two things, just like you can feed on either chapati or rice, depending on where you grew up, right? So the same thing is you can give bacteria lactose or glucose. Yeah, one is chapati, one is rice for them. So normally, if you are in glucose, so this lac operon is turned off, yeah, because you don't need it. There is no lactose there. But if you put in lactose, now what happens? Huh? Oh, there's a trick here. Don't be so into rush to tell me your answer. When you just put lac will get turned on.
For metabolizing the lactose, lac operon will turn on and it will activate the three genes. By using that, you can metabolize the lactose.
Yeah. So what you have is you had glucose to begin with. Life was happy. It is so glucose is kind of like going to McDonald's, right? You just show up there, bring your wallet out. Either you have some rupees in that or a credit card, right? All you need to do is, hey, give me that, Maha burger with whatever that stuff is, right? You come out, eat and well. The lac operon is kind of like you are a farmer. You need to put the seeds there irrigate it, wait for the harvest, and then do all the hard work before you can make your chapatis. Yeah. So the lac operon, it requires that extra effort. So there is a repressor called the lac repressor that binds to the operator sequence, so it prevents polymerase from binding. Yeah. When there is no lactose there. If you throw in lactose, now it binds to the repressor and makes it inactive. So when the repressor is inactive, now polymerase can happily go and bind to the promoter sequence and transcribe. Yeah, and it makes into these 3 genes, LAC-Z, LAC-Y, and LAC-A. One of them is called beta-galactosidase, and that's very useful thing in many aspects of biology. But anyway, so now what happens if you have both glucose and lactose in the same cell? Hold on. If everybody talks, then nobody can understand who is saying what. You go. Glucose will be used first, right? And that's a very smartness, right? Because if you can go to the McDonald's, why go to the farm and do all the hard work? Yeah. So the same things here. And the molecular basis for that is there is a so-called secondary messenger in the cell, yeah, called cyclic AMP, which is controlled by the level of glucose. High glucose means low cyclic AMP, low glucose means high cyclic AMP. High cyclic AMP, it binds to its own protein called CAP/CRP, and it is there to, remember I said over here? So our lac repressor is the situation here. When lac repressor binds, no transcription. If you put lactose, it inactivates that, so polymerase can bind this. The glucose is our this situation. When you have low glucose, so that means high cyclic AMP, it now binds to that cap protein. It now helps this to bind to otherwise weak promoter sequence and allow transcription. Okay, the other is a famous called tripoperon. Yeah, what's unique about tripoperon? Anybody from here? Hold on. So in the case of tripoperon, so there is a situation kind of like this, except that here this repressor is active to begin with. Yeah, it can bind and inhibit. In the case of trip, so it comes at 2 levels, the regulation. So one is, it is called aporepressor. Aporepressor meaning it is not active repressor yet, but can be made active. So the trip acts as a, if it binds to the aporepressor, it makes it repressor, so it inhibits it. So, and there's also what is called attenuation, and that is to make the system respond very fast, because there's a coupling of transcription and translation and to turn off the operon. And finally, have you heard of, there's a bacteriophage called lambda? Yeah. So lambda has 2 decisions to make, either to kill the cells or not to kill the cells. One is called lytic cycle, other is called lysogenic cycle, right? So the question is, how does it get decisions taken? Pardon? So there are these 2 proteins. One is called CRO, CRO. If CRO wins, it goes into the lytic cycle. If on the other hand, C1 wins, it makes it go to the lysogenic cycle, yeah, and so on. All right, so this is what happens to our famous bacteria, where we understand all the details about it. So now let's just go to a bit about the eukaryotes. So in eukaryotes, unlike in bacteria, so in bacteria there is only one RNA polymerase. Yeah, in eukaryotes, I hope you have watched this movie Slumdog Millionaire, right? And one of the questions in that movie is, who are the 3 musketeers? And I won't ask you who wrote, Alexander Dumas is the one who wrote this long time ago. Did you all watch Slumdog Millionaire? So during this part you were just sleeping, right? Yeah, you were just focused on that girl being saved. Yeah, So anyway, so these are our 3 friends in eukaryotes. So they are dedicated to certain type of genes, and the question is, what experiment led to the discovery that there are 3 polymerases in eukaryotes? Remember, in the 50s and 60s, somebody somehow figured something out that said there are 3 polymerases. So I'm telling you the facts, but I'm asking you, so what led to that discovery? And anybody from this table? So memorize this structure. It's called alpha-amanitin. So alpha-amanitin comes from this little thing, mushrooms that you eat a lot, I hope, on at least once a week, if not more. So alpha-amanitin binds to the active site of RNA polymerase, which we now call as RNA polymerase II. Yeah? And this is put after, so ignore this part. The experiment is that it is done with and without RNA polymerase. Yeah? No, with and without alpha-amanitin. And they looked at the transcription of these 3 genes, mRNA, ribosomal RNA, and tRNA. And the question is, what can you conclude from this experiment? Anybody from this table? You are testing transcription of 3 genes with and without alpha-amanitin. Yeah. Right. So what was found was that there is no signal or no RNA made for this guy. Where did I put? On the other hand, this is happily transcribed, this is happily transcribed. So this led to that there is some genes which can be inhibited or their transcription by just putting alpha-amanitin. But there were other genes which were not affected. So the idea was, what's happening? And that led to the discovery that, oh, that's because there's more than one RNA polymerase in eukaryotes. Yeah? And anyway, now there are ways to inhibit this also and so on. So anyway, so these are our 3 amigos. Now remember our friend sigma? Yeah? Now, if you look at eukaryotes, yeah, or humans, do you think there is a sigma there? And the answer for that is no. Yeah, so just say it no. But whatever sigma is doing there, that also needs to be done in eukaryotes. Yeah, what that thing is, you still have to tell the polymerase to go to the right place and start transcription. So that is done by what we call as GTF, or general transcription factors. So just one is shown over here as TF2D. So it is a complex of TBP plus other things called TF2D. It binds to this part, and now transcription will start over here. Yeah? So there is no sigma factor in eukaryotes. Yeah? But there are proteins who do conceptually what sigma does. It is telling RNA polymerase to start transcription over here and do the transcription. Now, so the in eukaryotes is more complicated. So there is a what is called, so this is RNA polymerase II. Have you heard of what is called CTD? Carboxy terminal domain. So what happens is that polymerase binds here, starts transcription, and as it moves along, you see this little thing over here. That's called our CTD, or carboxy terminal domain. So what happens is that in eukaryotes, there are other things that must happen, which do not happen in bacteria. And those are capping, polyadenylation, and splicing. And don't take this lightly, because this also happens in plants. And if you are ever going to do crop improvement, we need to understand all of those things. Yeah. So the point of this is that this CTD acts as a landing pad for all of these things, something that you will need for capping. So when the RNA is started, it now binds here and puts a cap. Then, oops, it moves further, and now the splicing machinery comes and binds. It moves further, and now the poly A tail machinery comes and binds. And the point of this is that as the RNA starts to come out of the polymerase, these are there to take care of all of these post-transcriptional processing events. So that doesn't exist in bacteria. And in eukaryotes, the two things come into play. One is called enhancer sequences. So anybody from here, what is an enhancer and what do they do? Right. So this is our polymerase, and what they found was there are these sequences which can activate a gene, but from far away distance. Yeah. So these were called enhancers, and the property of so how many of you have never heard of enhancer before? Everybody heard of enhancers? Okay. So the enhancers are, what's that? You're telling your friend something. You have never heard of it. Okay, so now you have heard, right? So enhancers are that they can act from a distance, and they work in these two forms. So number one is that they can act in an orientation independent manner. So remember the promoter sequence? Promoter works only in one direction. Yeah? But enhancers, suppose this is enhancer, yeah, in this piece of DNA. If you have this, it works. If you can do and flip it around, it still works. That will not happen with the promoter sequence. Second thing is, suppose this is your gene, transcription starts over here, this is an enhancer. So it can work by activating here. If you take this guy and move it long distance away, it will still activate it. So it works in a distance independent manner, and it also works in a orientation independent manner to activate genes. Now, life is not so simple. Even with enhancers, they bind to what we call as activator sequences. So activator binds to the enhancer and allows transcription. But now there are certain factors, they do not bind to a DNA sequence, and yet they activate. So we call them as co-activators. Yeah? So they do the same thing, and that is that suppose, so ignore this for a moment. This enhancer binds to activators and now will help polymerase to bind to promote and start transcription. But there are these factors, they do not bind directly to the DNA, but they bind to proteins which are bound to DNA, and then they activate transcription. And this is a big area of gene regulation. And the same thing applies to here. There are some proteins, they do not bind to a specific DNA sequence, but can repress genes. So they are called co-repressors. And so on. So in bacteria, what I call gene expression as an on and off switch. Yeah? Lac operon, you turn on and off. Trip operon, you turn on and off. Lytic lysogenic, you can go either this way or that way. Yeah? But now, that is not good enough for eukaryotes, including animals and plants. Why? And therefore why new solutions are needed.
Last number of non-coding sequences are there, and huge size of them.
Pardon?
Multicellular.
Multicellular, right? So the point is that bacteria is a single cell, right? And it can respond to its own off switches. But in our body, there are trillions of cells. Same thing in the plants. There are billions of cells. But now the thing is that there are some genes which are turned on and off, but other genes are expressed at different levels. So the question is, how do you fine tune? So the word is called combinatorial control. And what is combinatorial control? Our friends in this table. Combinatorial control. So combinatorial control is that suppose there is a promoter X, Y, or Z. It has a bind. It has these enhancers of F, G, B, C type, and A, B, C, D, E, and so on. Yeah. So now, what you find is that some enhancers are unique to one gene, other enhancers are shared by multiple genes, and what you have is each of these promoters has binding sites for more than one thing. Yeah. And the more than one thing allows is that depending on, let's just say we are looking at humans. So we have liver, kidney, brain, intestine. So what you'll find is that you are not just turning on and off, you are changing concentration of a given RNA in different cell types. The question is, how do you accomplish that? So by doing this multiple regulators, yeah, you can fine tune. So depending on how much of which protein is expressed in a given cell type, it will bind to each of these factors, right? And that will lead to change in concentration. So unlike the so I don't know how, so imagine there are two kind of switches, right? One is you turn on and off. There are other which you slide that you can dim. So by doing this command and control, you can do this gradient of how well the light is in the room, right? Remember that sliding switch? So depending on how far you go up or down, you can go from no light to full light and everything in between. Bacteria cannot do that. Lac operon is either on or it is off. There's no mechanism to grade that, okay, 25% on or 50% on. But in human body or in the plant cells, we can do that depending on in what ratio you express these proteins that bind to these sequences. And one key about combined control is that suppose you have 10 genes to control, yeah? Then you need 10 regulators, right? Regulator 1 for gene A, regulator 2 for gene B, and so on. So that you have one dedicated controller for each of these genes that you are trying to regulate. Yeah? But now suppose if 1 and 2, they regulate A and B. If 1 and 2 can dimerize, yeah, form a dimer. So now they can regulate a gene 11, right? But if 1 can talk to 2, suppose 1 also can talk to 3, 4, 5. So now by these different combinations, you can create a new heterodimer combination and uniquely control a new genes. So the point here is that you can control lots and lots of new genes uniquely without increasing the number of these regulators. Yeah? How do you economize use of regulators? By a limited number of regulators, you can combine them smartly and activate genes in a way that you want. So I'm almost done for this part of it. So this is to get to the sense of, so in the last few years, there are what is called the super enhancers. And who has heard of super enhancers? If you have heard of, raise your hand. Super enhancers. So that will be part of your homework. Yeah? So anyway, so super enhancers are that there are these genes which are of this type and they are controlled by super enhancers. And what I'm going to do is I will somehow get you this 1 or 2 papers that I have identified, and tomorrow or on Friday you are going to present. Yeah, and we'll send you the instructions as to how to do it. So what I have done is I have selected a few papers, yeah, where you will be standing here, and what we'll do is we'll make you into a group of 3 or 4 people. Yeah, it will be assigned a research article. Yeah, and the point here will be that in about 5-7 minutes, yeah, what is the executive summary of that paper? Yeah? Okay. So what time is it now? And when did we start? Pardon? 11. And I'm talking for an hour and 15 minutes. So I think it's time for a break. Right? Okay. So why don't we have a few minutes break? And let me do of that break, use 5 minutes to write down this and then tear that piece of paper off